buckle up and put your analysis helmets on because it's going to get crazy. Okay, so some of you may already know me. I'm Carter. I guess I introduced myself earlier. Yeah, I'm Carter, if you guys forgot. So I've been working at the Allen Institute for about 3 years, and I was hired specifically by Jerome Lecoq, who's in charge of the OpenScope program, specifically for this to make this data book. So let me just talk about OpenScope for a little bit. So OpenScope it's a grant NIH funded program that Allen Institute offers. Ben mentioned it a little bit earlier, and essentially the mission is to facilitate reproducible and open science, right? Collaborations between many institutions, because the Allen Institute now has a ton of infrastructure for really high quality data, and all these experts are there. We put a lot of expertise into producing multimodal, high quality data, and we want to maximize its usage, right? We don't want just one or two teams to use it. We want to have many people use it and make it as public and available as possible. So the thing about OpenScope is it's on a yearly cycle, so anybody can apply, scientists, non-scientists, and NIH fully funds it. So the idea is we leverage the Allen Institute's internal data analysis pipelines and data acquisition pipelines to provide data and run an experiment for the applicant, for the awardee. And then we provide the data to them for 1 year. They have private access to it. And then after 1 year, we have a commitment to release the data publicly to anyone on Dandy. So Ben mentioned we already have 11 data sets up there. And right now, I think there's 3 or 4 pending that will be released very soon. In the next year anyway. And I just wanted to encourage, because normally Jerome would be presenting here, but he didn't make it this time. But I just want to encourage you guys to all apply if you have a really good idea. You want to leverage the Allen Institute's faculties and run your own project. So I can post some more information in Slack later. But I'd encourage you guys to look into that. So we've been doing this for 3 cycles now, and we have a ton of data sets. So Jerome hired me to help facilitate help work on ways so we can facilitate the reproducibility of our data. So without further ado, my project is the OpenScope DataBook. You guys already know a lot about Dandy, so I don't need to hit you over the head with that. So the background 3 years ago when I started was we now had this data. We had it in this NWB format. It was still maybe less familiar to the community at the time, and the ecosystem wasn't as mature. And there's many, many common types of analysis on the data that we were collecting, but there wasn't any really clear centralized pillar of the community that helped facilitate the analysis and helped provide reusable code that would be reusable on many different data sets. And we anticipated that there'd be a lot of different NWB data sets coming in. And so we wanted to add code that would work, for the most part, out of the box on any of our NWBs to do all the things that Ben mentioned earlier, right? What we can do, aggregating data between many different labs and data sets. So the specific goals we set out to solve with the data book were performing these common analyses that I mentioned really quickly with a push of a button or two. We wanted to provide the code and the ability to access and process our data. Importantly, we wanted to educate the users on the data, the experimental details and the technical details. There's a lot of different expertise and skill levels among you guys, for instance, and a lot of the information is it's kind of opaque online for a lot of other data sets. So we wanted to sort of start breaking through that convention. So explaining things like the principles behind electrophysiology, optophysiology, how the probes work, how the NWB format is actually structured, things like that. We want people to be we want it to make it easy to generate and share our figures. And ultimately, all of this is basically just to facilitate the proliferation of our data, like I mentioned. So we did it with a web-based repository of Jupyter notebooks using a platform called Jupyter Book. Basically, it's just a web page that consists of a couple chapters, and each chapter has many Jupyter notebooks in it. It's hosted for free on GitHub Pages. And another important detail is that it's launchable to different compute platforms. So instead of just providing the code as like a static document, you can launch them on DandyHub, which you guys have all used now. And you can also run them on Google Colab, or you can download them and run them locally. So some of the content, and this is not an exhaustive list, but these are the highlights. A lot of it is kind of replicating what Steph presented earlier, like actually how to use NWB files, download from Dandy. Like I mentioned, actually the details behind NeuroPixel and 2Photon. So a lot of sort of basic visualizations, just explaining what the data are. And then like other things that you guys might be familiar with, like receptive field mapping, opto tagging, a lot of details about the Allen Institute's quality control for EFIS. And then there's also some higher order stuff like TCA, CBRA, which was shown this morning, and like GLMs, for instance. So I would highly encourage you to look at all of these. If you're unfamiliar with any of the analytical techniques in there, you feel like it would be good to know, or if you sort of want to dive into some of the data that you're less familiar with. So like I mentioned, it's launchable on multiple computational platforms. This slide is a little bit outdated, so I would, Binder does not really work, so I'd recommend Google Colab if you just want like an individual notebook, but I think DandyHub is probably your best option. I'm going to do a demo later, but recently Dandy made some changes, so we're still working through getting those super automated. And like I said, yeah, you can also run them locally. A new addition to the platforms that we're including in the DataBook is called CodeOcean. This is a computation platform that the Allen Institute adopted about a year and a half ago. And we're very close now to getting it, specifically the DataBook publicly accessible on CodeOcean. So I think soon, probably in the next year or two, if you guys are following any of the Allen Institute releases, there are going to be a lot more material that's going to be released by the Allen Institute on CodeOcean. Specifically stuff like our actual packaging pipelines for all of our data sets we're releasing on Dandy. So all of the code, basically from the moment it comes off of the rig, or the experimental rig, goes through Spike interface, and then all of the processing and all of the packaging that it goes into, we want to make that available to the public. So that's a goal of ours. We're not there yet, but the DataBook being released on CodeOcean is a start. Access to data, you guys already know how to do this now. Okay. So it's easier for me to explain just by showing you guys. So let me just pull it up here. So to find the DataBook, you can just search. Actually, don't just search DataBook. That's something with stocks. OpenScope DataBook is what you want. Don't get confused. You can go on GitHub here. So this is the repository. One important detail is that we have a citable DOI page on Zenodo. So we treat the DataBook kind of like it's either a textbook or an analytic resource. So like a publication, essentially, right? So having citable DOI is one part of that. Another part of that is having an authors list. So this is an automatic authors list that the website has. It's pretty nice. And just as one reflection of OpenScope's commitment to open science, anybody who commits to this repo, which is openly available, will automatically get added to the authors list. And there's sort of different views that you can see. Yeah, and so we've had a number of many external contributors providing their notebooks and their analysis and findings to the DataBook, like Guillaume, who's recently arrived. And I just want to encourage you guys all, like if you do, use some of this code or you analyze code data off of Dandy and you think you produce useful findings that would be really broadly useful to the community, the DataBook, I would highly encourage you guys to reach out to me to try to add that to the DataBook. And the process essentially just goes through GitHub PR, but we're doing our best to leverage the modern sort of tool stack in the ecosystem to try to basically manage like an actual scientific review process. So typically we have a PR and then all the code gets reviewed by several internal and external people. Essentially to help push the part of the ecosystem that sort of promote this new wave of open science and treating it really rigorously, but also, leveraging modern tools to do it efficiently. Okay, let's see. So this is the website, as you guys can see. This basics chapter was pretty much covered today already, so it might not be that useful to you, but it might be a useful reference if you need to go back to some code for streaming NWB files or accessing them off Dandy. One tiny note. I don't know if did you show how to get the path this morning? Okay, so if you find a Dandy set you like, say, you type in the Dandy set ID here, and then this path, some people have questions about how to get this usually. If you go on Dandy and you go to Files, you click on a file, this I will lead you to the asset metadata page, and then it will take you to the path for the file. So anytime you want to download or stream a file, this is what I would recommend doing. Okay. So what are some example notebooks here? Let's go to LFP. So this is local field potential. It's electrophysiology. And this notebook is just a good demonstration of how you can just kind of plug in a notebook and then reproduce like really key or simple findings. So in this case, it downloads, this is from a visual coding data set. It downloads the two files, and there's a couple of fields that would need to be changed if you change the NWB, like the names of the stimulus here. But for the most part, this notebook should run with very little modification. So you can pull out, in this case, this is the stimulus table, which just shows the events, the visual stimulus shown to the mouse throughout the duration of the experiment. You can pick particular stimulus times. This is kind of arbitrary, but I just selected frame 150, or excuse me, when the orientation is 150. And this subsample is a part of the period, a part of the session, and interpolates. You specify like a event window start time and end time, and then it produces these nice event-aligned LFP responses to the stimulus. So you could do this with any of our Allen Institute data sets that have this data in there, and you'll get something like this, which is really great. It just sort of expedites the process. So as you guys are starting your projects, I would recommend sort of playing around with notebooks like this and our data sets, just to sort of start off as a good launching off point. Another one I'll go over briefly is receptive fields. So this does essentially the same thing, but this is also electrophysiology. Gets the stimulus information, and it gets unit information from particular neurons, and it pretty much automates the process of pulling out the different degree orientations, and then generates these really great receptive fields from our data. And you can just, like I said, you can just plug in various Allen Institute data sets and then maybe change a handful of fields and it will work well. I won't spend a lot of time today going over these, but these are super useful analyses for like higher order stuff like I mentioned. So Zebra was shown to you guys this morning. So this code will be really useful if you want to leverage Zebra. And this notebook I will be going over tomorrow. And then, yeah, something like GLMs or tensor composition analysis would recommend. I do want to spend a little bit of time highlighting this chapter. So this chapter specifically was made so that we can actually present our OpenScope data sets to you guys. Like we kind of already mentioned, it's really hard to just see a data set on Dandy and know enough about the experiment that you need to know to have really productive analysis, right? It takes a lot of time. It takes some digging or maybe contacting the owner of the data set. So this was designed to mitigate that for our data sets. So these notebooks were written mostly by the external teams that the Allen Institute has worked with for our OpenScope data sets. And we released them with the data so that people have really clear access and sort of understand the experimental design quickly. So let me go over, let's see. I think I'll go over this one here. So we released our sequence learning data set a few months ago. And this goes over the experimental design. It's, I didn't write it, so I would encourage reading it. I wouldn't be able to do it justice. But basically this describes a summary of the actual experiment, like how we split up the mice, this is a table of the metadata that I was actually pulled from Dandy. So it's a summary of the files and a summary of the different subjects. Downloading the file, you can see this is the overall NWB. We show if I were to run this notebook, you'd see these as interactive videos, but these are the actual stimulus videos shown to the mouse. Same thing as previously, you select stimulus times. This is an overview of the ROIs extracted from this session. So these are the individual cells from the 2-photon field of view. And then, this is for the whole session, but this is what the data actually look like. We also have the mouse running. So I think pretty much all of our OpenScope data sets include the wheel running data and the eye tracking, which may or may not be here. Okay, so eye tracking is not here, but we also include the gaze using the DLC eye. We also have this code here, which sort of gives you an overview of the actual order of events that the stimulus was shown here. So this is just a slice of the session, but you can see the sequence of how stimuli were shown here. And there are other session types. This is a repeated. So for most of the session, the stimulus is in a perfect pattern. So that's why it looks a little fuzzy here. And then there's a random section here. So, and you can see we have the running data and the fluorescence throughout the whole session. So they're very rich in the data. And I think like if you wanted to focus on a project that asks questions about the mouse's attention, using the running data would be really useful. And the eye tracking as well, of course. The final details of this notebook is it also just really quickly gets you that event-aligned response information here. So this gets you that, and you can see there are pretty clear responses in the fluorescence. And you can sub-select cells based on their responsiveness. And one other note is, this doesn't show it, but if you wanted to do something more ambitious with the fluorescence, we also release the files that have the raw, like 2-photon movie, basically the recorded images from the field of view for the entire session. That's very large, so I wouldn't recommend doing that locally. I'd recommend doing that on Dandy Hub. And if you ran this code, you'd also be able to just literally watch the actual brain during the session, which I think is pretty cool. Okay. I don't think there's any other details to show you for the data book itself. Let me go back here. Lastly, I just want to reiterate what I already said, which is we would really like to encourage people to contribute to the data book, and we want this to grow as the ecosystem grows. So as new analyses become common, as new tools get developed in the ecosystem by you guys or your colleagues, we want to help integrate those and help disseminate that information through the data book. So we're very open to different types of contributions. And like I said, we have a very rigorous review process, and everyone who contributes becomes an author. The OpenScope project has many, many collaborators that are not fully listed here because they could not all fit on a slide, but I did want to acknowledge that the databook was not just produced by me, and OpenScope is made possible by the hard work of many, many different contributors. So I want to acknowledge some of them here. All right, are there any questions? Yeah.
So there's 11 OpenScope data sets on Dandy,
Mm-hmm.
and pages on the databook for each of them.
Yeah.
Are there suggested experiments or follow-up analyses that come with any of these data sets, like for people who are looking for projects?
That is a great question. Some of them have that. I don't know which ones. I think this one at the bottom here mentions, let's see, other things you can find or exercises for the reader. There's some suggestions here. I don't think most of them have that though. But yeah, I would encourage reading, like if one of them does seem like it has data in there that's interesting to you, I would encourage just reading the content on Dandy, the description, and then maybe skimming the notebook, and it might give you guys some really good ideas. Yeah.
Are you open to contributions that use other data sets, or do you want all this data to be Alan?
No. So, I mean, that's one thing. OpenScope, we're very passionate about helping many people disseminate, their findings and facilitating collaboration. So, I guess selfishly, it would be great if you used our data. But if you have your own data set that you think is high quality and broadly useful, then I think that would be a great contribution. Yeah.
Yeah, can you tell something more about the process of applying to OpenScope and how it works? You have an idea of something that, if you understand well, the experiment is carried out at the outlet.
Mm-hmm. Yes.
And then how does it work for the analysis? Is the person that submits the project involved into, like, is it project specific and kind of changes?
Yeah. So the things that the Allen Institute does is we collect the data. I think we write the stimulus script for the awardee, but the awardee has a lot of control over the stimulus and basically decides what it's going to be. But we just actually write the script typically. I mean, the rules aren't very strict here, right? It's highly interactive and collaborative. The other things we do is we quality control the data. I mean, it frequently happens, right, where there's various issues. The mouse doesn't learn properly or the data just has issues. So we sort of cull down the actual data set to be the ones that we think are high quality. And then we do a lot of pre-processing to the data, like a ton of pre-processing in ways that are mostly lossless, right? Like you don't lose a ton of vital information in what we do. And then we package them into NWB for the awardee and release them as NWB. So that's sort of the specification of the project. But the awardee is fully responsible for any real analysis. Like I do analysis with a lot of our collaborators, but ultimately it's the analysis would be their responsibility.
The data set is available, right? So it doesn't go to the awardee first, and then it's.
So the data set, so typically we release the data sort of iteratively, right? I mean, as we discover problems with the processing and things. So we'll do like initial releases shortly, like within, I mean, it varies by project, but lately we're able to get our data packaged, like a rough draft of our data really, really quickly, like within a month of starting collection. But we usually, the whole process takes about a year of collecting and processing and packaging. And then we do an official data release where we say, by this point, typically the awardee has had many, many months to do an analysis on the data. But this is formally where we say your analysis period begins, and they have 1 year before we release it publicly. Yeah. As far as applying, I can find you that information. If you look up, OpenScope, you'll be able to see information here. Yeah, there'll be an apply button somewhere around here. I'll send it when I find the link.
What kind of experiments can you apply with? Is it all neuropixels or 2D and mice?
So we do neuropixels and 2-photon are the main thing. I'm pretty sure we only do mice. I am not sure if we have a new microscope called SLAP2, and I'm not sure if that modality would be available, but it might be. But primarily neuropixels and 2-photon imaging, yeah.
What kind of behaviors or tasks can you work with?
This, I think, is pretty unspecified. I think it's flexible. There's certainly, basically you're at the mercy of the committee who chooses the projects to see how scientifically viable they think the experiment is, but there's no specific limitations.